Okay. That's good. Primary immune response.
I wasn't here, so I don't know. I'm just
Okay.
asking.
So we've
No, we got through the ability of the immune system. Yeah.
7.
Two paper to give.
So we are here. Are we here? No, yes, maybe. Primary? Okay, let me go back. Okay, so we are here. Alrighty. Okay. All right. All right. Any questions? All right. Any questions? Okay. When is your last exam? Exam 4? No, you have the date for that. April 20. Not exam 4. You have to take exam 4 on that day. Yeah. What is the date, please? April 30th. What's the, yeah, April 30th. So you, Tuesday, April 30th. Okay, let me write that down. So exam 4 is Tuesday, April 30th. 30th. Okay. And what is today? 18th. Okay. And your final in lab is next week? And that is what day? 25th. So 1 week after that. Okay. All right. All right.
Are we still taking that fifth exam on the same day?
I was thinking maybe a different date. Yeah. I was thinking as this optional cumulative. Maybe a day before this. Before. Not after. Not after. Not Monday. Thursday. Final? Oh, shoot.
We can't do it on
After this?
Yeah, after that,
After that, Tuesday. Let me look at my schedule. I can't really say, so let me look at my schedule. Maybe. Okay? Let me see. All right? Yep. Let me look at the schedule. Okay. That's true. Yep. Yep. Remember, we are finished with the lab a week before. Yep. Week before. Yep. So let me see what I can do. Okay. All right. Yep. All righty. All right, but exam 4 on the 30th, Tuesday. Okay. I think it's at 10:30, not at 11. 10:30. Yeah. 10:30. Yeah. Cumulative? Exam 4 only, not cumulative. Okay. Yeah. 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 All right. Okay. Okay, let me look at my schedule. Okay? All right. Sure. Any question on natural active or passive immunity? All right. Let's see if we can finish up this chapter today, and then we'll move on to the last topic of this class, which is antibiotics. We'll do that next time. All right. Majority of the exam will come from 16, 17, which is immunity.
17 is in here?
This is 16 and 17.
It's just labeled chapter 16, and you have all the information.
It's labeled immunity, but this is both 16 and 17.
Oh, okay.
You swear no?
Okay. Yeah.
No, you don't need
All right.
Yeah, must give us another type of.
All right. Okay. Duality of the immune system. Okay. Or you can simply say at what level the B lymphocytes are involved and at what level the T lymphocytes are involved. Or you can say functions of B lymphocytes and functions of T lymphocytes. Or another way to say, okay, humoral immunity and cellular immunity. Humoral immunity, which is also called the antibody-mediated immune response. This is an immune response that protects you if the pathogen or the toxin or the microorganism is present in body fluids. Body fluids. Okay? Blood, but not the cellular component of the blood. Okay? If the pathogen is in RBCs or WBCs, okay, like PY-vax, Toxoplasma gondii, or viruses, they have invaded your cellular component of the blood, then this type of immunity will not protect you. Then the other type of immunity, which is called the cellular immunity, that will protect you. But if the pathogen is in the blood, okay, like Trypanosoma gambinis is parasite, but not intracellular parasite, this type of immunity will protect you. Okay? How does it protect us? By making antibodies. Okay? So B lymphocytes are directly involved. Humoral immunity protects us by making antibodies. Antibodies. Protect us from bacteria, bacterial toxin, and viruses. But viruses are obligate intracellular parasite. I just told you viruses, okay, we are protected from viruses by means of cellular immunity, right? How come it is right here, viruses are here? when do you think humoral immunity will protect us from viruses? When viruses goes through lytic cycle and they cause the death of the cell, and the cell dies and they come out in body fluids, then this type of immunity will protect us. But as long as the virus cell, this type of immunity has no effect. Okay? When the host cell dies and the viruses come out in body fluids, this type of immunity will give us protection.
The virus will continue intracellular.
Pardon me?
The virus will continue.
If it is in lysogenic phase, hiding inside the cell, no protection. Okay.
So even with the humoral, it can.
No detection. Yep. Cell-mediated immunity. Okay. This is the one that provides us protection from intracellular parasites. Okay. How? They don't produce antibodies. Instead, they make antibody-like substances called lymphokines. Lymphokines. And both lymphokines and antibodies are called together opsonin. Opsonin. And opsonins are substances that do what? Enhance phagocytosis. Enhance phagocytic activities. No antibodies are involved. This type of immunity protects us from intracellular bacteria, other parasites, and viruses and parasitic fungi. So those are your two types of immunity. Antibodies. Antibodies. Yeah. Or you can also simply call them death markers, markers of death. Anything that enters into an immunocompetent host is called an antigen. True or false? Anything that enters into an immunocompetent host is called an antigen. False. Here's the criteria for an antigen to initiate an immune response. Okay, in order to initiate an immune response in an immunocompetent host, is called antigen or immunogen. Now, if this antigen will initiate a bad response, by the way, are all antibodies good for the host? No. What is not good? Which antibody is no good? Yeah. Autoimmune, or one is called IgE antibody that causes allergic reaction. That is no good. And that is found in our body in the least concentration. Least concentration. So not all antibodies are good for the host. Okay, autoimmune antibodies are no good. Okay, and those are called allergens. Allergens. An antigen that causes the synthesis of IgE is called allergen. Okay. Exactly. Yep. Mm-hmm. First criteria for an allergen, okay, molecular weight. Minimum 10,000 Dalton. Let's say if it is 8,000 Dalton, what would you call it? Hepten, H-A-P-T-E-N, an incomplete antigen is called hepten, H-A-P-T-E-N, hepten. It must react with another hepten to become a complete antigen. So two heptans can react together to become a complete antigen. Your body must be able to process it. Okay? If it cannot process it, it will not produce any antibodies. Here's a good example right here. If you take a bacterium called pneumococci, pneumococcal antigen, if you inject it in an adult human and a little mouse, guess what? Little mousey is going to produce much higher antibody titer than an adult human. Why? because the little mouse has many more enzyme to process it, break it down into smaller pieces than an adult human. Human has very few enzyme to process pneumococcal antigen than a mouse. So processing is very important. In other words, a human can break down pneumococcal antigen into, let's say, maybe 4 pieces, the whole pneumococcal molecule. A little mouse can break it down into 100 pieces, and each piece will produce an antibody. So processing is very important. Okay? Foreignness. Foreignness does not necessarily mean that it should come from outside. Foreignness means it should be foreign to your immune system. I'm not sure if I mentioned this to you or not, but there are certain proteins, okay, that are foreign to your own immune system. Lens of your eye, not the eye, lens of your eye is foreign to your own immune system. Breast milk of the female is foreign to your own system. Sperm of the male is foreign to your own immune system. All these proteins, if they are accidentally exposed to your own body, a female can lose the ability to lactate. A male can become infertile. Okay? You can lose your eyesight if the lens of the eye becomes exposed to your own immune system. So foreignness means, okay, foreignness to your immune system. Epitopes means foreign. Epitopes means antigens that are found on these proteins. So foreign means antigens that are foreign to immune system. Okay. Complexity, very important. How many types of proteins exist in nature? 4. Proteins exist in nature in 4 different shapes. Primary structure, secondary structure, tertiary structure, and then finally quaternary structure. Quaternary is hemoglobin. If you take 4 mice and inject each of this protein in a mouse, 3 and 4, which one have the strongest immune response? And one of them will have no immune response at all. This mouse, no immune response at all. No complexity, no antigens at all. This mouse will have little immune response because whenever the protein change, they cross, that's where they make their antigens, or they are called epitopes, antigens. So this one will have a little bit of immune response. This mouse will have stronger immune response. This one will have the strongest immune response. Okay? They look like mice. Yeah. Okay. Cats. Okay. All right. All right. All right. Handout. Everybody has a hand? No? Okay. On the back of this handout that you're going to get is the next topic we are going to talk about is antigen elimination curve. And you have a graph that I'm going to draw. This one, this kind of ugly, but I'm going to draw it again right here so you can follow me. Right here. Antigen elimination curve. What happens to an antigen when it enters into or when it encounters the immune system for the very first time? And this is going to be on your exam. Actually, I have merged this with something else. Okay, so we are going to practice this as soon as I'm finished explaining it to you. Okay. first, this is time. First, we have time in minutes. And then in days. Five, 10, 15, and then 20 minutes. And then days. Five, 10, 15, 20. And then this is, on y-axis, we have concentration of antigen. Concentration of capital A, small g is abbreviation for concentration of antigen. 100%, 50%, 0%. Okay. Within about 20 minutes, 90% of the antigen is removed from the system, okay, through phagocytosis. So this line that I'm going to draw, it represents down. This is phagocytosis. By the way, I'm going to 1 with a degree sign. This represents that this is person's primary response. This person has come in contact with this antigen for the first time. Primary immune response. Okay? If I put 2 with the degree sign, that will be secondary immune response. So within about 20 minutes, 90%, what 90%? Could be 95%, approximately 90%. Phagocytize, gobble, gobble, gobble. We talked about phagocytosis last time, right? Right? Okay. Remember, what was the last step of phagocytosis? Antigen cell, the phagocytic cell became what? Antigen presenting cell, right? Okay. So after this, now this phagocytic cell now becomes antigen presenting cell. After this, there's a long silent period, long catabolic phase. For about 15 days, this right here. This is processing phase or catabolic phase. Processing phase in which your body is getting ready to make antibodies. Okay? What I'm talking about here, we will do this diagram later, but this is what's going on right here in this processing phase. Okay. Okay. Here's phagocytic cell. It became antigen presenting cell. Antigen presenting cell presents the antigen to the factory that makes the antibodies. Okay. And after about 15 days, what are made? What is made? Antibodies. Okay? This step that I'm skipping, I will come back to that later. But after about 15 days, antibodies are made. And what is the function of antibody? Opsonization, remember. They attach to antigen. Okay, remember that. So, remember, about 10% of the antigen is still somewhere escaping in your system, right? 90% is gone. So, right here, after 15 days, antibodies are released. After the catabolic phase, antibodies are released. And what is the last step? What does it say? Remaining antigen, or you can say remaining 10% antigen. Remaining 10% molecule. Antigen molecules are what? Opsonized. Opsonized meaning antibody-antigen reaction takes place. These antibodies, when they are released, what do? They actually hunt the escapees, right? And then they react with the antigen and 100% elimination of antigen. In this phase, antigen, antibody reaction take place, and then 100% elimination. And then phagocytosis. %, elimination of the antigen. Make sense? Kind of? Please? All right, take one more time. Okay. So this is where you eat the hamburger, contaminated hamburger. 90% is eliminated by phagocytosis. 15 days. Okay. Your body takes about 15 days, sometimes a little longer. Antibodies are released. Those antibodies, they hunt down scapees. Antibody-antigen reaction takes place, 100% elimination. I'm going to draw a line and tell me what happened here in this case. What does this represent? What happened there? Pardon me? Go ahead. Secondary, very good. Secondary immune response. To the same bacteria, you went to the same shop, you ate the same dirty hamburger again, sorry. But this time, you have the memory cells. Your catabolic phase, your response time is much shorter. This time, boom, not 15 days, 7 days. Okay. So this is your secondary response, shorter time. Very good.
So on your initial exposure,
Yep.
once it goes, the phagocytosis goes down to 90%, you're still infected for another 15 days until,
Yep. Mm-hmm. Yep.
will you still show symptoms? I mean, I guess it depends on.
It depends on your immunity, how much you consumed and all that. Sure. Yeah. Sure. You may be asymptomatic. You may not get sick. It depends how much you consumed. Okay. Yeah.
Is there a point where you can spike back up? Where the infection would increase?
Increase or decrease?
Increase after the phagocytosis.
After phagocytosis?
Or
Within 15 days, if you go back and consume another burger, yes, sure. You can go and sick. Sure. Absolutely. Sure. Yeah. All righty. All right. What is the origin of these cells, B lymphocytes and T lymphocytes? Where do they come from? All blood cells, they come from bone marrow. Their mommy is this right here, hemocytoblast. Okay. What does blast mean? If you tell someone, oh man, you're a blast. Immature. Next time you tell someone you're a blast, you be very careful. What you're really telling that person is, you're so immature. Exactly. Blast means immature. That's what it means. Embryonic. That's what it means. Okay? Blast. Hemocytoblast. Blast, when it enters into, this is the one that you need to remember for the exam, lymphoblast. Don't worry about all the others. Lymphoblast, when it enters into thymus, it becomes what? T cells or T lymphocyte. Cyte is cell, mature cell, cyte, T lymphocyte. If lymphoblast enters into GALT, gut-associated lymphoid tissues, spleen, appendix, lymph nodes, Peyer's patches that are in small intestine, specialized tissues in small intestine, they become B lymphocyte. So let's look at these organs one more time. Sorry, go ahead.
The ruboblast, all these,
Yep.
if they go into the thymus, will turn into T lymphocyte.
No. Rubriblast, okay, yeah. Only lymphoblast, okay, will become thymus, okay, in T lymphocyte. Rubriblast will become RBC. Yep. Pardon me? Yes. Peyer's patches, appendix, lymph nodes. This is the only one. They are T cells mature. No T cell, no thymus, no T cells. We already talked about bursa fabricus, right? No? Yeah. Where the B cells were first isolated. B stands for bursa fabricus. Right? Yeah. All righty. Now let's talk about how antibodies are made. We have already talked about the first two steps. We need to just look at the third step. Antibody response or antibody synthesis. How antibodies are made. First step, microbial antigen is processed by phagocytic cell first. Non-antigenic parts are released by the phagocytic cells. Antigenic parts are kept. Antigen is presented on the surface. Now the phagocytic cell is now called antigen presenting cell. Why is it called antigen presenting? Because it presents the antigen to the B cell. Now, if the antigen is T dependent, the antigen, yes, sir? Thank you very much. Is Taylor here? Taylor? I don't have anybody by this name.
Okay.
I'm sorry.
All right.
No problem. Anybody by the last name Taylor? I don't have anybody by the last name of Taylor. Anybody? All right. So, this antigen presenting cell takes the antigen and stimulate the B lymphocyte. That's called stimulation of B lymphocyte by antigen presenting cell. In other words, they literally fuse together. That's called antigen stimulation of B lymphocyte by antigen presenting cell. When they fuse together, let me go to the diagram. No, first let's read it and then we'll go to the diagram. When they fuse together, they split into two types of cells. One is called plasma cell and the other one is called memory cell. Okay? In primary immune response, during primary immune response, plasma cell produce antibodies. And during secondary immune response, memory cell produce antibodies. All right, let's look at the diagram now. Okay. Okay. Here we are. Phagocytic cell, antigen presenting cell. One step that you may want to draw here, which is not here. If this antigen is T dependent, I want to draw an arrow here this way, right here. Draw here. And draw another cell and label that cell as T helper, TH cell. TH, T helper cell. Okay. And you may want to just make a note. Okay. T dependent antigen. Okay. If T dependent antigen, okay, then just make a little note. If T dependent antigen, okay, and then draw back arrow here. Okay. So you have an arrow going here, another cell here, and label that cell as what? T helper, and then draw the arrow here. Then you have 3 cells, APC, B cell, and T helper cell. Okay. You may want to make a little note here. T independent. This process is T independent. Independent. There is no helper cell. So T independent. T independent antigen. So antigen presenting cell, take this T independent antigen to B cell. This is antigenic stimulation. They merge together and they split into 2 types of cell, plasma cell, memory cell. Here is one with a degree sign, primary immune response, plasma cell produce antibodies. 2 with the degree sign right here, memory cell produce antibodies. Make sense? Right, exactly. Yeah. Okay. Yes.
So if the T cell is here instead of the T cell goes with it, the same thing happen?
Same exact thing. The 3 of them will split into 2.
Okay.
The 3 of them will split into 2. Okay. If it is T dependent, the 3 of them will fuse and they will split into 2. Okay? Yeah. If it is T dependent. Okay? All right?
So T dependent
Right.
will combine and then break into B.
No. If it is T dependent, antigen presenting cell, okay, and B cell, all 3 will fuse together to split into 2 types of cells. Yeah. Instead of only antigen presenting cell and B cell splitting into 2, 3 cell will fuse to split into plasma and memory cell. Okay? Combined. Yeah.
And that's, okay.
The end result remains the same.
But in T dependent, there's just the T helper cell, just
Right. 1 extra cell.
stimulation.
That's right. 1 extra cell involved. Yep. Alrighty. Primary immune response versus secondary immune response. This graph is going to be on your test too. Primary versus secondary immune response. The second graph on the back of the diagram that you have. Yep. Title graph. Yep. Primary immune response versus secondary. Long processing phase. We have already seen that. Rest, I'm going to actually draw this and compare the 2. Low antibody titer, the concentration of antibodies that are made in primary immune response is low. Lesser avidity, what does that mean? Avidity towards antigen. Bonding between antibody and antigen is not strong. The bonding between antibody and antigen in primary immune response is not strong. Like in primary immune response, like sometime even when you put the cuffs on criminals, they can escape, just like that. In primary immune response, when you put the cuffs on criminals, they can escape. So in primary immune response, even with the antibodies on, okay, bacteria can run away. Okay, yeah. So bonding is not strong. Antibodies that are made in primary immune response, they belong to class IgM. There are 5 classes of immunoglobulins. Okay? And in primary immune response, they belong to class immunoglobulin M, IgM. Decrease in antibody titer is rapid. Their lifespan is short. They do not stay in the bloodstream for a very long time. Okay? As compared to secondary immune response. Secondary immune response or booster or anamnestic immune response, short catabolic phase. Why? Memory cells. Memory cells. High antibody titer. Higher avidity, the bonding is strong. I am a strong or an avid fan of Cubs, right? Avid fan, have you heard? Strong fan. Avid fan. Higher avidity, strong bond. Antibodies are mostly IgG, and they last for a long period of time. Now let me draw and practice. Okay. 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 Let's first just practice simple and then complicate it. Antibody titer. Antibody titer. Time. Okay. Blue line indicates primary immune response. Red line indicates secondary immune response. Okay. Letter A indicates long processing phase. Okay. Antibody titer is, you can see right here, is low. Okay. The letter B indicates what type of antibody? IgM. IgM, primary immune response. Okay. Letter Y, okay, indicates what type of antibody? IgG. IgG. Stays in the system for a long period of time. Decline is gradual, slow. Decline is sudden. short life, like this. Mhm.
This might be a statistic answer. What kind of question this is? But why do we use those terms IgM, IgG, and IgE? Don't we use those terms to define immunity?
Those are Greek terminologies. I'll find out what they stand for. I did remember them a long time ago, but I forgot. Yeah. Do you remember what they stand for? Okay, yeah, I'll try to find out.
IG stands for
Yeah.
immunoglobulin.
Yeah, but G and M,
Type M, G, E, A, B,
EA. Okay. I,
E, M, G, one more time. E is for hypersensitivity, and
Yeah.
IG, A is in secretion, M and G is in the blood for the
Okay.
infection. As you mentioned,
yeah.
M is primary, G is secondary and longer.
What they stand for? Well,
I think when you look at the structure of immunoglobulin, it has it's made of heavy light arms or heavy chains and light chains. It has something to do with the structure of it. If you
this
look
is
at
how
the
they look
protein,
like.
Yeah.
Yeah.
Yeah, if you look at the structure, it comes with the alpha, gamma, O. A is for alpha, G is for gamma, E is for epsilon. It's those Greek words.
Greek
Yes.
words? Okay,
And
thanks. Okay.
it relates to the structure of immunoglobulin itself. Because like, I don't remember it's IgG or IgM. It's made of 5 arms.
That's M, the heaviest. M,
Yes.
yeah. M.
One of them is made of 2, one of them made of 1. It's just the arms and the structure.
Okay,
But it's
yeah.
alpha, gamma, epsilon. I forgot this.
Those are fraternity names.
Yeah, and those relates to
But you may be right, but I'm going to double check. Yeah, you sounds right, but I'm going to double check. Okay? Yeah. All right. Good. Yeah. All righty. So this makes sense? X? I just made it up. Yeah. What letter would you like? No, I just made up those letters. Nothing really. It's just it could be C, D, E. Yeah, it could be any letter. Yeah, it's just this letter X actually represents something, which is the start of secondary immune response. That's what it represents. The start of secondary immune response. Letter A represents the start of primary immune response. That's what they represent. Yeah, could be any letter. Okay. Yeah. Okay. Pardon me? Catabolic phase, as you can see, very short, very long. Yeah. All right. I'm going to practice. I'm going to draw. Should I draw another one to practice with you now? Any question before I draw another one to practice? All right. All righty. Okay. Which letter or letters, okay? Which letter or letters of the graph indicate the start of secondary response antigen A? Which letter or letters indicate, represents the start of secondary immune response to antigen A?
C.
I hear Y, I don't know, but I hear C, and I hear C and X. Third choice is C and X. Um, D. Okay, correct answer is that's it. There's only one letter. I said start off. Start off.
But, okay, you said C, but when secondary antibody is present, I mean, the attack is so immediate,
Yeah.
in a matter of seconds.
Sure. Yeah.
But C, it looks like there is no response, it's close to zero.
Start off. Start right. It could be right as soon as you have memory cells. It can start right after, but it cannot be before the.
Yeah.
Cannot be before. Yeah.
So the first one, the XY, that's the secondary response
To a different antibody. Different. Yeah. It is
In
secondary.
order like that on the chart,
Yeah.
it's going to go, it's
Yeah. This is a secondary response
to one something.
to something else before. Yeah. Yeah. Before. Not to this one. Yeah. Yeah. Okay.
Okay, my question is, so what, I'm sorry, the same YX is now,
X is a secondary response to a different antigen.
oh, because you put the blue in the,
Yes.
okay.
Yeah. No. Yes. Which letter or letters represent the synthesis of IgG? Very good. D and Y. And which letter or letters represent the synthesis of IgM? Very good. Which letter or letters represent the synthesis of IgE? I haven't told you yet. Haven't, yeah. Okay, good. I'm going to make it more complicated, yeah. Okay, all right. Because you understand. So let's see. Good. All righty. All righty. Let's see. All righty. All right. Enough letters? Okay. All right. Okay.
What do you want to know?
Okay. Okay. Which letter or which set of letter or letters represent the synthesis of IgG?
What is 1, 2, 3?
Choices. Choices.
So that's,
These are your three choices.
one of them is the answer of your question.
Yes.
Oh, yeah,
2 is correct.
yeah.
Very good. This is secondary. This is secondary right there. Short. IgG, IgG. Very good.
So it's just a shorter.
Very good. Shorter catabolic phase. Shorter catabolic phase. Long catabolic phase. Shorter catabolic phase. Higher titer. Okay. So I merged the two. This is antigen elimination curve, and this is antibody synthesis. Yep.
On the antigen elimination curve,
Yes.
secondary.
Second. Yeah, the big B.
So that's when the antibodies are released.
Exactly.
So how does that correlate with the question? I guess I'm a little thrown off the question itself.
Okay. Yeah, during secondary immune response, this is where after 7 days, the antibodies that are made, they are IgG. Yeah, yeah. And then here you make IgM. Yeah, yep, right.
I maybe nitpicking here, but shouldn't the phagocytosis of the initial, or of the secondary be shorter in time than?
No, because antibody, yeah, that's true, you're right. You're right, because of, yeah, you're right. Because of,
So that
And-
line should be inside the blue.
yeah, it should be smaller. Yeah, yeah, that's true, yeah. Thank you. You're right. Pardon me?
It's just a punishment if we get it from the test.
This will-
I guessed it, because I kind of look at it, but it's punishment.
But
Happy?
at times on the bottom, wouldn't the red line have to come before the blue line and go down?
You're absolutely right. I will fix it. Before test, I'll have it perfected. You're right. Yeah. 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 Okay, go ahead. Sorry. Question?
IgG is made on the decline of the
Second. IgG is made in the secondary immune response. Okay. Let me fix it. So I don't want to confuse you, those of you who are taking pictures. Better? Oh, I know. I know. Sorry. Finally.
So on the and IgG's made it kind of lot.
No problem. Yeah. 1 second. Somebody?
What you had up there before, I did capital B and D, the IgG is made at those points.
Okay. Antigen elimination curve or? Okay.
I think the AG concentration curve.
Okay. Right here. This is the original one, David, right here. This is the original one. without any complication. This is primary immune response right here. Primary long catabolic phase, and the antibody that we make is IgM. Okay? And then in secondary immune response, yeah, sure, right here, IgG, like that. Okay? IgG. Now, if we merge this one with primary immune response, okay, this is, we make IgM here, and we make IgG here. If you want to merge them, you can kind of superimpose them. That's what I did right over here. Yeah. Okay.
Just basically, when you look at the shorter lines, where's IgG is going to be always shorter.
IgG, what shorter?
On the secondary, it's going to be a shorter.
Catabolic phase. Exactly. Shorter catabolic phase. Okay. Yeah. And right here, this one too, shorter. IgG, always. Okay. Yes.
The first diagram, the IgG should be not after the
This one, right here.
the top one.
This one.
Yeah.
No.
Line should not be after this.
After this.
Yeah, because first we will have exposure, then the secondary. line.
No, but the processing phase is short.
It should be short, but
It's happening before the primary, so it looks like the secondary is in reaction
before
to something
primary.
else. Like the first one that you asked us, you had
Yeah.
like which letter or letters are in response to
Yeah.
the.
Well, days, let's say if this is day 1, day 2, 3, day 4. By days, if you go day by day, it could be day 7, and this is day 15, so this is day 7. Okay? Yeah.
It's just a
So,
different chart setup than the first
yeah.
one you did, which was
right. Yeah. Yeah. Makes sense? Maybe?
Those one make sense. This one, or
No.
this one not going to be on the test. Are you going to give us more?
That is going to be on the test.
This
Yes.
younger jungle?
Yeah.
So you're gonna give us more to show.
We will practice. We'll practice more. All right? Yeah. All right. All righty. Structure of antibody. Okay. How much time? 10 minutes. Plenty time. Plenty. All right. Structure of a typical antibody. Antibodies, they belong to the class, they're all proteins, and they are called immunoglobulins or gamma globulins. They all look like a letter Y, and they're made up of 4 polypeptide chains. 2 of them are heavy, okay, and 2 of them are light. I'm going to draw my own diagram. This is a bad diagram, okay? And these 4 chains are linked by disulfide bonds. Okay. Let's draw antibody structure. Okay. the two heavy chains, okay. First, let me just draw everything, then we will label them. The small chains are called L chain or light chain. The long chain is called H chain or heavy chain. And both L chain and H chain, they have this red part that I have drawn. This part is the one that gives uniqueness or specificity to the antibody, this red portion. And this is called variable region. Variable region of the antibody. Variable region. Okay, let's say if there are 5 million antibodies in group IgG. This part will be the one that is different in all of those antibodies. And this blue part is the one that is called constant region. That will be same among all those 5 million antibodies. Like enzyme, if you look at enzymes, which part of the enzyme makes it specific? Active site. Active site. That makes it unique, right? Okay, so this is the part that makes the antibody unique. The rest of the part is called the constant region. Constant region. Constant region. Okay. Let me make it a little bit more room so I can draw my bonds. In between these antibodies, they are held together by disulfide bond. Two sulfur bonds. Sulfur, 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 sulfur. That's the typical structure of an antibody. Two small chains, two heavy chains, and disulfide bonds. I can make that a little longer. Okay. Most of the antibodies made up of C region, the blue part, which are the same for all antibodies of the same class. Both the H and the L chains, they have sections located at the end of the Y's arm called variable regions, which bind to the specific antigens. We stop here today. Next class, we are going to finish this chapter and start the next chapter, which is antibiotics. Okay? Good day.